Hello, my name is Trina Ganguly, and the date today is February 11th, 2023. And today I will be discussing the movement of macromolecules through the cell. In specific, we will be going into how steroid hormones, protein hormones, and intracellular hormones travel through the cell. To begin with, the steroid hormone I'll be discussing is progesterone. So in this image, we are observing a flowchart or an imagery flowchart of progesterone synthesis. And we can see where one is labeled as the nucleus, where DNA is created, and then DNA gets transcribed into RNA, which then gets translated into mRNA. And then the mRNA moves out of the nucleus to the ribosomes, which are bound along the rough endoplasmic reticulum, which is labeled as 2. Then from there, the cholesterol goes to the Golgi apparatus, which is labeled as 3. And then from there, it goes over to the outer mitochondrial membrane, which is labeled as 4. And when the cholesterol gets to the outer mitochondrial membrane, it gets greeted there with specific enzymes and proteins, which will then let it into the inner mitochondrial membrane, which is labeled as 5. In specific, the translocation of cholesterol from the outer to the inner mitochondrial membrane is mediated by protein-protein interactions of cytosolic and outer mitochondrial membrane proteins. And so once it gets inside the mitochondria depicted as label 5, the cholesterol then gets metabolized into a steroid, which is called pregnenolone. And then the steroid is then catalyzed into progesterone. In specific, the side chain cleavage enzyme CYP11A1 gene converts cholesterol to pregnenolone, and so in order for the catalyzation process to get activated and occur, the pregnenolone goes back and forth between the intracellular areas labeled as 6 and the smooth endoplasmic reticulum labeled as 7 to exchange their intercellular particles. Then after the catalyzation process, pregnenolone is turned into the steroid hormone progesterone, which then goes to bind itself to the cell membrane, which is labeled as 8. So it can exit the cell membrane since it is lipophilic, and then progesterone passes through the plasma membrane to get DNA information by binding to nuclear receptors, which is labeled as 9 and 10. And lastly, it travels through the bloodstream so that it can travel throughout the body and eventually get to the endometrium to prepare for the potential of pregnancy after ovulation, and that is shown with 11. Next, I'll be discussing the protein hormone insulin. And so in this image, we are observing insulin synthesis. And so first, DNA for insulin is created on the cell nucleus of chromosome 11. Then the DNA is transcribed into RNA and then translated to mRNA. And prepared for transport. Next, when the mRNA leaves the nucleus and attaches itself to the ribosomes bound to the rough endoplasmic reticulum, which are also known as fixed ribosomes, then those bound ribosomes translate the mRNA into protein, and the insulin proteins, where the signal peptide from mRNA, which is also known as proinsulin, is cleaved by a signal peptidase to yield proinsulin. And the proinsulin then undergoes folding in the rough endoplasmic reticulum along with the formation of 3 disulfide bonds in preparation for being transported to the Golgi apparatus. Then the folded proinsulin is transported from the rough endoplasmic reticulum to the Golgi apparatus, where proinsulin enters immature secretory vesicles and is cleaved to yield insulin. And so the secretory vesicles leave the Golgi apparatus and go to the plasma membrane, where it binds with the plasma membrane and then secretes insulin into the extracellular space. Then when the insulin is in the extracellular space, it travels to find more insulin receptors that are along the cell surfaces throughout the rest of the body. And finally, I'll be discussing the intracellular protein actin, which is a cytoskeletal protein and is a major protein constituent of the microfilaments. And so in this image, we are observing the synthesis of actin-binding proteins. First, similar to the other processes as discussed, DNA for actin-binding proteins is created on the nucleus, and then the DNA is then transcribed into RNA, which is then translated into mRNA and prepared for transport to the ribosomes. And so when the mRNA leaves the nucleus, it attaches itself to the free ribosomes within the cytoplasm, and these free ribosomes then synthesize the globular actin proteins so that they can be used within the cell. Then the globular actin proteins polymerize into small actin aggregates containing 3 actin monomers to form actin filaments, which are long strings of actin-binding proteins bound together to create actin cytoskeleton near the plasma membrane of a cell. And here are my references. Thank you.